Hello guys, my name is Gustaf Lay. I'm from FK Biologi with name 23010400018. Now I will explain about DNA transcription. Let's see. Before that, I will including how is the transcription. Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of this is transcription unit. This portion of the DNA will be used to various RNA. Upstream of the transcription unit is the data box. An adjacent region may also be involved. Several complexes known as transcription factors are required for successful transcription. The first is TFIID, the largest of the general factors. A component of these factors, TBP, binds to the DNA, using the data box to position TFIID near the transcription initiation site. Other transcription factors, including TFFI and TFFB, then attach. This complex prepares the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now energy must be added to the system for the transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then system and RNA template from the strands of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates, and the newly formed strands of RNA is released. Now I will explain the transcription of DNA. DNA transcription is the process by which the genetic information contained in DNA is rewritten into messenger RNA or mRNA by RNA polymerase. This mRNA then exits the nucleus, where it acts as a basis for DNA translation. By controlling the production of mRNA in the nucleus, cells regulate the rate of gene expression. Where is the DNA transcription happened? This process occurs in the nucleus of both prokaryotic and eukaryotic cells. At this stage, each DNA nitrogen base is encoded into an RNA nitrogen base. How is the process transcription DNA? The DNA transcription process can be divided into two main stages. The first, initiation, second, elongation, and third, termination. Initiation. Transcription is catalyzed by the enzyme RNA polymerase, which attaches to and moves along the DNA molecule until it recognizes the promoter sequence. This area of DNA represents the start point of transcription, and there may be several promoter sequences in DNA molecule. Transcription factors are proteins that control the rate of transcription. They also bind to the promoter sequence with RNA polymerase. Once bound, the promoter sequence RNA polymerase unwinds part of the DNA double helix, exposing the base on each other and two DNA strands. Elongation. One DNA strand, the template strand, is read in the 3 to 5 direction, 3' to 5', and thus provides a template for a new mRNA molecule. The other DNA strand is called the coding strand. This is because the base sequence is identical to the sequence mRNA except for the replacement of the base element with uracil. RNA polymerase uses incoming ribonucleases to form a new mRNA strand. This is done by catalyzing the formulation of phosphodiester bond between each ribonucleotides using complementary base pairs A to U, T to A, C to G, and G to C. Bases can only be added at the 3' end so that the strand extends in the 5' to 3' direction. Termination. Elongation continues until RNA polymerase finds to a stop sequence. At this point, transcription stops and RNA polymerase releases the DNA template. Pre-translation processing of mRNA. The mRNA that has been transcribed up to this point is referred to as a pre-mRNA. Processing must occur to convert it into the mature mRNA. This includes 5' restriction. Restriction describes the additions of a methylate guanine cap at the 5' end to the mRNA. It presents essential for recognition of molecule by ribosome and for protecting immature molecules from degradation by RNase. Polyadenylation. Polyadenylation describes the addition of a poly A tail to the 3' end of an mRNA. The poly A tail consists of several addition monophosphate molecules. This helps stabilize the RNA, which is necessary because RNA is much less stable than DNA. Connection. Splicing allows the genetic sequence of one pre-mRNA to code for many different proteins, thereby preserving the genetic material. This process is sequence dependent and occurs within the transcripts. This involves removal of inserts non-coding sequence via splicing excisions, joining exons coding sequence by ligations. At the end of the transcriptions, the mature mRNA has been decreed. It acts as a messaging system that allows protein translations and synthesis to occur within the mature mRNA. There is an open reading frame, or ORF. These regions will be translated into protein. It is translated in blocks of 3 nucleotides called codons at the 5' and 3' ends. There is also an untranslated region, or UTR. It is not translated during protein synthesis. That's all my explanation about DNA transcription. Thank you. Hopefully this material can provide useful lesson and understanding.